Hello friends, welcome to another interesting video. So in the series of discussion, we are discussing the basic concepts about cell cycle and cell division. We had these discussion in our previous videos. So in that we have talked about what is cell division, why cell division should happen, okay, and what are the different phases of cell division. And friends, this discussion is going to be a continuation of the previous video where we had talked about the inside of a nucleus, because we believe that the nuclear division, that is karyokinesis, which can happen either via mitosis or meiosis, before which we are supposed to know what is there inside the nucleus, right? So the previous video we had discussed about that, what is DNA, what is chromosome, why DNA is getting condensed into a chromosome, etc., etc. So kindly look back to those discussion and then join us here. So in this, as I said, I'm going to continue or discuss a bit deep into the discussion what we had done last video, right? So with that, welcome to this discussion. Friends, last class we have talked about, that is the DNA, which is a double-stranded helical structure. Hope you will be aware of that. So one runs from 5 prime to 3 prime, other is going to run from 3 prime to 5 prime, right? So double-stranded helical structure. If I am going to unwind this DNA, I am going to have, say, two parallel strands like this, 5 prime, 3 prime, okay, and this is going to be 3 prime, 5 prime, right? Both are same, but both are wound together, both the strands. Here both the strands are unwound together, right? In that I am going to take one particular strand, say 5 prime to 3 prime strand, okay, 5 prime to 3 prime strand. This is made up of, you know that it's going to be made up of nucleotide ranging from N1 to N. So N is nothing but nucleotide. So it's going to start from N1 and it's going to end with N. The N value might vary depending upon the length of the DNA. So this is going to be a summary of what we had done in last discussion. Friends, I'm going to take to a different aspect now, but anyway related to this, that why is this DNA and what is the importance of DNA? That is what the main purpose of today's discussion. So before that, friends, let's get into the another discussion, something called as protein. Okay, we will have some basic discussion about protein and get back to continue with our previous discussion that we are in. So proteins are the one basically it is going to be a chemical constituent or a biochemical constituent of a cell, protein, right? Any food that you take, it is obviously going to be rich with protein, like fish, meat, etc., etc. Though the other biomolecule like carbohydrate, fats are present, but protein are going to be the major constituent, as our buildings are made up of brick, though other components are also a part of it, right? So this is protein, and proteins are made up of amino acids, right? Proteins are made up of amino acids. So friends, you have 20 amino acids which combine together to form how many protein? The answer should be, I can say, millions and millions of protein. So I don't have a definite number here to say how many number of proteins are known, okay, to us, but I can say the numbers are definitely higher. Okay, so from 20 amino acids you get millions of protein. The question is, how this 20 amino acids, okay, make a significant number of protein is what the question that we have now. Okay, say for example, let you take protein P1 and you take protein P2. From the million protein, we take protein P1 and P2. See, these amino acids are going to combine in a way, say A1, A2, so it goes till AN. And here also you have A1, okay, and it goes till AN. Friends, this N value Okay, this is what is called as a peptide chain, because you see the amino acids are getting linked, okay, through a peptide bond. So you call this as a peptide chain, and this n value, say if it is 51, here the n value, say it's going to be 62. So this indicates the length of the protein or the length of the peptide chain, which varies from protein to protein. So this is one, okay, significant difference that you see between P1 to P2, right? My question is just 20 amino acid, but you get variety of protein. What is the reason? So that's what I am discussing here. And this is one case. So I can say the length of the peptide chain. Okay, this is going to be one reason. The second reason can be, say for example, if two protein, the n value is going to be same, so both are 55, 60, so there is no difference in that, then the sequence might different. Okay, so the other one is amino acid sequence. So the specificity of a protein lies with either the length of the protein or the amino acid sequence. So these two what makes a protein specific. As I always say, you have 0 to 9 digits, right, with which you have n number of telephone number that we have. Some are 8 digits, I mean yours might be 8 digits minus 7 digits. So even in case if both of our numbers are say 7 or 8 digits, then our sequence is going to be different, numerical sequence is going to be different, and that make my and your phone number unique. Hope you get this point, right? So with this, we'll go back to the previous video. Friends, let me take this nucleotide, say a stretch, say N1 to N200. So what is this stretch? I can say this is going to be called as a gene, alright? I'm going to call this as a gene. So what is a gene? A gene is a stretch of nucleotide which is seen in DNA, yes, which is going to code for a specific amino acid, specific protein, that is P1. It is going to contain information for the specific amino acid P1. What information? Just now we have talked about, okay, how many amino acids this P1 have and what is the sequence of the amino acid. That's what, okay, that the information that gene is going to have. Okay, say in case if you have 10 protein, I might have 10 gene, right? So this is G1, this is G2, this is G3, I might have, okay, 10 gene, and all gene pile up together to form DNA. Say for example, as a human, if I have millions and millions of protein, I would expect millions of gene, and all gene will pile up to form DNA, right? So this is how, okay, things are arranged. So I can say nucleotides will all pile up to form a gene will all combine to form a DNA, right? And I say one DNA cannot accommodate more number of gene, many number of gene. So in that case, for example, let's consider 10,000 proteins. Let me assume 10,000 protein an organism has. So it need 10,000 genes. So for example, a single DNA can accommodate 1,000 gene, okay, 1,000 gene, which means that it needs another 9 DNA. So this is one DNA which is going to accommodate 1,000 gene, but total required is going to be, total gene is going to be 10,000. So this organism need totally 10 different DNA to accommodate the entire 10,000 gene that is needed for that has the information for the 10,000 protein, right? And these DNA, as per our last discussion, will condense to form, okay, chromosome. Okay, hope you people get my point, okay, until here, right? And friends, this is going to be the reason why different organism has got different number of chromosomes. Say, for example, humans have got 23 chromosomes. Okay, humans have got 23 chromosome. Can say C1, which goes till C23. So it has got 23 chromosomes. Okay, 23 basic types of chromosome. As I said, each chromosome will be in pair, that is father version, because we get one set of chromosome from father, one set of chromosome from mother. So you call this as a C1F, and the same pair will be seen. This is called as C1M, like that's C2F. This is going to be C2M, like that 23. C23F and C23M. So totally 23 into 2, since it is pair, we have 46 number of chromosomes, right? So depending upon my summary here is that depending upon the organism, the protein number will vary. Depending upon the protein, the gene number will vary. Depending upon the gene, the DNA number will vary, and the number of DNA will be exactly equal to the number of chromosome. Okay, so this is the idea here. Friends, let's take this particular chromosome alone. That is let's take C1F and C1M. And you know that this is going to be a centromere, which I am highlighting it here with a different color, centromere. This is called as a chromatid. This arm is called as a chromatid, right? Fine. Now what happens is, you know, there is a phase in interphase which is called as a S phase, synthetic phase, and we have seen that in synthetic phase the DNA replication is going to happen. Okay. Likewise, if the DNA replicates, what happens? This is called as a monad chromosome, monad, M-O-N-A-D. I call this as a monad chromosome. Friends, now in S phase, due to DNA replication, right, this is going to become a dyad, right? It's going to become a dyad. You call this as a dyad. Now here, the chromosome, okay, chromosome number remains same, because always you count the number of chromosome by counting the number of centromere. So centromere is going to be one. The centromere is going to be, you know, in both the cases there's going to be the same number of centromere. So chromosome number remains same, but the chromosome concentration is doubled, which means that if you have C in pre-synthetic phase, before S phase, if the chromosome concentration is C, then it is going to be after S phase, after this, you know, becoming dyad, it is going to become 2C. So this is one important thing. And friends, the DNA replication, what is happening? That is this process too. Okay, this process. Let me mark the centromere here. So this is the C1, C1F, and it's going to be C1M. Okay, in this you call this, I call this as A, I call this as B, I call this as C, I call this as D. I can call this A, B, okay, both as sister chromatid. Okay, I call this as a sister chromatid, and I can also call these two as sister chromatid. Okay, sister chromatid, because both are exactly same. Whatever is there in A, that is also going to be in B, because your A is a copy of B through DNA replication. So both are called as sister chromatid. Okay, ademari C and D is also called as a sister chromatid, right? Now when you compare A and C, okay, first of all you call this as this is C, this C1F, okay, and C1M. This is C1F, C1M, both are same chromosome but of different parental origin, one from father, one from mother. So you call this as a homologous chromosome. I am writing here. Okay, you call this as a homologous chromosome. You can call, say, C1F, C1M as homologous chromosome, C2F, C2M as a homologous chromosome. The idea here is the same chromosome but of different parent, you call it as a homologous chromosome, right? And the chromatids of homologous chromosome is going to be called as a non-sister chromatid. I'm going to write here, these are called as non-sister chromatid. So what is the difference? See, friends, for example, if you see that the C1F is going to have, you know that chromosome is going to have genes, because DNA is going to have gene, the pile-up of gene nucleotide sequence, there is a gene is what is called as a DNA, and container of DNA is what is called as a chromosome. So obviously the chromosome being a condensed version of DNA should have gene, and you call the point of that gene is what is called as a locus here, right? Fine. Now let's say I'm writing it here, the C1F, C1M. First gene is going to be height. Here also is going to be height. And second gene is going to be some X gene. Here also X gene. Third gene is going to be Y gene, is also going to be Y gene, because the gene profile of both homologous chromosome C1F and C1 is going to be same. And whatever gene you see in C1 will be different in C2. Okay, you hope you get my point, right? Now friends, I have taken a very known gene, that is height, because you would have talked about this height gene in your Mendelian experiment previously, because Mendel considered the height of Pisum sativum plant, right? So this height gene might have, okay, two different expression, that is one that is tall, other one is going to be dwarf. Okay, so I can call this as capital T, okay, this is called as small t. The capital T is one phase of this gene and small t is going to be one phase of this height gene. So we call this as an allele, okay, A-L-L-E-L-E, right? So my point here is this H gene here, either it can be capital T or it can be small t, or it can be either capital T or it can be small t. So I can have capital T homozygous, or small t homozygous, right? Or capital T capital T, which is heterozygous, okay, and this is called as homozygous. So my point here is, okay, the non-sister chromatids, that is ANC or AND, BNC or BND between homologous chromosome might have same gene profile, but allele might be same or might be different. But in short, both are allele profile between homologous non-sister chromatids are not same, they are going to be different. And this exchange of non-sister chromatid between homologous chromosome is what we call it as a recombination, which we will be discussing in the part of meiosis, right? And apart from this, see, you have a gene profile of C1F, C1M. If you are going to take C2, as I said, whatever gene that you see in C1 will not be seen in C2 or C3, right? So this is what I am trying to convey, okay, the idea that is required for us to understand what is happening in mitosis and meiosis. So without these understanding, okay, mitosis, meiosis will not be as interesting as it is, right? Hope you get this point. In case if you have any doubts, okay, kindly get back to us. We'll be happy to clear, and we will continue our further discussion in another video. Okay, thanks for watching.